so I have nothing to disclose, and would like to thank the organizers for giving me the opportunity today to present our data here. So despite nearly 50 years of pharmacological as well as psychosocial research in schizophrenia, the field has only improved marginally. And while we do use antipsychotics quite successfully, most of them have difficulties in application and implementation of those. So if a psychiatrist is faced with a new patient, he will for sure treat him with an antipsychotic, but at that stage he won't know whether this patient is going to respond, at what time is he going to respond. If he doesn't respond, should he be switched to a different antipsychotics, or may he pull out and discontinue the treatment? So failure of using an antipsychotic, failure of successful treatment of an antipsychotic in first episode psychosis is one of the major factors that has been shown to impact the long-term prognosis. And as such, at the moment, there is no satisfactory biological marker to predict which patient with the first episode psychosis will be responding to what treatment. So there certainly is a need, a pressing need for defining biomarkers to predict treatment response, and also to the sensitivity to side effects in first episode psychosis. Inflammatory pathways recently have been shown that they offer a new area for biomarkers of treatment response. For instance, IL-6 and IFN gamma have been linked with poor treatment response, but findings so far have been mixed, and as much as they are looking promising, further research is currently needed. So for those who have missed this morning's Optimise symposium, just to give you a quick overview, Optimise stands for the Optimization of Treatment and Management of Schizophrenia in Europe, and is one of the rare studies that tests treatment algorithms. So the aim of the Optimise study was to improve treatments for patients by first optimizing the current treatment, but also by identifying new treatment strategies through a series of biological experiments, be it MRI studies or MR spectroscopy, proteomics, and as I've learned this morning, cytokine assays, as well as metabolomics. So the Optimise trial is a cross-collaborational study between more than 20 centers across Europe, as well as Israel and Australia, and it is a large project funded by the FP7 European Commission. More than 500 patients have initially been recruited with the first episode of schizophrenia, and these patients had minor treatment with antipsychotics beforehand. So the study's aim was to treat these patients initially with amisulpride as a first step to see how well they are responding, and then the non-remitters were either kept on the amisulpride treatment or were switched to olanzapine, and again, non-responders after the 10 weeks of treatment were then switched to clozapine as an early clozapine treatment. So where do we come into place? Our study was a proteomic study, so we wanted to generate a proteomic profile in these patients, in a sub-study, in a subset of these patients, to identify proteins that are differentially expressed between the patient group at baseline. And they were clustered by the successful treatment with the amisulpride after 4 weeks. We wanted to see whether we can predict treatment response in these patients, and ultimately identify pathways for new treatments. So as I said, we used a subset of those samples. So we used baseline plasma samples in patients who, after 4-week amisulpride treatment, were known to be either a non-responder or a responder. And we used our normal plasma proteomics machinery. So we depleted those samples by HPLC, depleting them of the 14 most abundant plasma proteins. And then we used equal amounts of each sample and tryptically digested them into peptides before they were run on the mass spectrometer, and afterwards they were searched against databases to identify the proteins behind it. So we found initially 464 proteins, but when we used stringent criteria, so samples could only be included in the study if they were identified in more than 70% of all samples in the study, we ended up with a data set of 228 proteins. And we then used ANOVA to test for group differences between non-responders and responders, and we're also adjusting for age, gender, and BMI at that stage. That left us with 25 proteins that came up to be significantly differentially expressed in our study, and Bonferroni correction then showed the pro-collagen endopeptidase enhancer to survive FDR correction, as well as the neural cell adhesion molecule and alpha-1 microglycoprotein to just not make the significance criteria. So everything else is seen as P smaller than 0.05. So we had quite a bunch of different identifications and therefore used pathway analysis. And when we looked at the results, we could see that complement and correlation cascades were the top pathway in our analysis, and 7 out of those proteins that came up significant could be assigned to this pathway. So here I've highlighted them, the complement proteins in red and the correlation proteins in green. And as Vanessa so nicely already explained, the complement cascade is a major effector in immunity, and it comprises of over 30 proteins that all interact closely together to fight an infection. And here I've highlighted within those complement cascades the protein that we identified at several stages within the pathway. So of course, no proteomics work without any independent confirmation. For this, we used a different set of those Optimise samples. Again, we worked with 30 non-responders and 30 responders. And we have chosen quite a number of proteins to validate for this work I'm presenting today. And for the study that we have finished, we were mostly interested in the complement pathway for sure, and very interested in C4A, as this showed the biggest fold change among all the proteins in the primary analysis. So we used ELISA to confirm the C4A finding, and as you can see, we were able to confirm the change of direction. So the top are the non-responders. So we reached 860 nanograms per milliliter in those samples in comparison to the responders, which had about 640 nanograms per milliliter, and that came up to a fold change of 1.33. But as you can also see, that just didn't quite reach significance. For these samples, we used ANOVA to correct for age, BMI, as well as the PANSS changes. So complement has been quite a topic, and there is compelling evidence that the genetic association in the big GWAS study conducted by SICA has identified the C4 locus in the major histocompatibility complex. And it has also been shown in this study that the genetic variation in this locus influences the mRNA level of C4a in particular within the brain. So this again highlights C4 as a potential biomarker. So to sum up, our data, it identifies the complement pathway, and in particular the C4a protein, in a preliminary study to predict treatment response at baseline within the Optimise. And this data complements findings by the GWAS study undertaken by SECA, who implicated excessive complement activity in the development in schizophrenia. And this is also supported by findings by our own group, where we described an upregulation of the complement pathway among those at risk of future psychotic experiences. So this data needs to be tied in with the bigger findings that have been undertaken within the Optimise study, and hopefully eventually will find some practical application that might hopefully impact the treatment. So I would like to acknowledge David Cotter and the team in Dublin that did the study, Patrick Dicker, who has done the statistical analysis on it, Jack Hackney and Kieran Wynn, who helped with the mass spectrometry, and Philip McGuire, who gave me access to the samples, Tom Pollack, who pulled out all the samples for the subset that I've been using, Rennie Kahn, who coordinated the entire Optimise study together with Inge Winter, but also everybody else in the Optimise. Thank you very much for your attention.
Thank you. And we have time for some